Welcome to the Stream of Knowledge. Today, we will discuss a very important topic, and that is culture media use in microbiology. What is culture medium? A growth medium or culture medium is a solid or liquid designed to support the growth and multiplication of microorganism or cells. There are two major types of growth media. Number 1. Cell culture. Cell culture use specific cell types derived from plants or animals. Number 2. Microbiological culture. Microbiological culture are used for growing microorganisms, such as bacteria or fungi. The first foremost question is why to grow bacteria. Bacteria have to be cultured in order to obtain antigens from developing serological assay for vaccines. Certain genetic studies and manipulations of the cells also need that bacteria be cultured in vitro. What are the general characteristics of culture media? 1. It must give satisfactory and rapid growth from single culture. 2. It should maintain pH during storage. 3. It should reasonably cheap and easily available. 4. It should maintain sterility. You should remember the common ingredients of microbiology culture media. This will help you a lot. The common ingredients of cultural media are 1. Agar. Agar is a solidifying agent. 2. Peptone. This is source of nitrogen. 3. Yeast extract, which is act as a source of vitamins. 4. Meat extract. This is a source of vitamins, carbohydrates. 5. Water. Water is used for the uptake of nutrients. 6. NaCl. NaCl is used to maintain isotonicity. Now there is another important question: what is agar? Agar is a polysaccharide that is derived from red seaweed. Its ability to melt at 85 to 90 degrees Celsius and solidify at 34 to 42 degrees Celsius make it ideal for culture media. It is used for preparing solid medium. It has no nutritive value, and not affected by the growth of the bacteria. It melts at 98 degrees Celsius and sets at 42 degrees Celsius. 2% agar is employed in solid medium. What are the types of culture media? You should remember this. Based on their consistency or physical state, there are 3 types of culture media. A. Solid medium. B. Liquid medium. C. Semi-solid medium. Based on chemical composition, there are 4 types of culture media. A. Simple medium. B. Complex medium. C. Synthetic or defined medium. D. Special media. Special media has further 6 types. 1. Enriched media. 2. Enrichment media. 3. Selective media. 4. Indicator media. 5. Differential media. 6. Transport media. Let's discuss types of culture media in detail. Culture medias on the basis on the physical state. Number 1. Liquid medium. In liquid medium there is absence of agar, or you can say a medium without agar. It is used for the proliferation of bacteria. For example, nutrient broth. Number 2. Solid medium. In solid medium there is 1.5 to 2.5% agar. It is used for the isolation and identification of bacteria. Example of solid medium are slant, petri dishes, plates. Nutrient agar, blood agar. Number 3 is semisolid medium. Semi-solid medium contain 0.3 to 0.5% agar. It is used for the observation of bacterial motility and preservation of bacteria. Example of semi-solid medium is SIM or sulfide indole motility. In our future lectures, we will learn an interesting and one of the most important topics. Types of culture media. So stay connected.
Thank you for watching. Do subscribe, like, comment, and share.